Good afternoon, dear colleagues. My name is Janneta Tcherkassova. I'm from UCT Research and Development. I'm from Canada. I'm very happy to be able to participate at the International Conference on Healthcare and Nutrition for the opportunity to present the results of a double-blind clinical study conducted with breast cancer patients. Breast cancer. So can you see the slides, right? Everything is fine?
Yes.
Okay. Breast cancer is the most diagnosed cancer globally. It accounts for 1 in 4 cancer cases among women and is the leading cause of death from cancer in women. The estimated more than 2 million new cases indicate that 1 in every 10 cancers diagnosed in 2020 was breast cancer. In many low and middle income countries, the cancer is more difficult to treat, is more expensive to do so, and is usually incurable. Compared to 1990, the global deaths of breast cancer increased by almost 84%, with 700,000 new cases in 2020. To resolve the growing breast cancer problem, it is critical that improvements are made to access to early detection of cancer, timely access to treatment and palliative care, as well as comprehensive data collection using through cancer registries. Breast cancer usually form a tumor that can often be seen on an X-ray or felt as a lump. Unfortunately, about 30% of early-stage breast cancers are impalpable tumors, which are difficult to detect. If breast cancer is missed at early stage, it's spreading outside the breast through blood vessels and lymph vessels and becomes advanced breast cancer. Survival rates for breast cancer are very high when the cancer is detected early and where treatment is available. Unfortunately, until now, about 50 to 80% of breast cancer cases are diagnosed at an advanced stage. Let's consider the epidemiological situation. Figure 1 presents a distribution of female cancer incidence for most cancers in the world. As we can see, Asia takes a leading place for cancer incidence, followed by the Europe and North and South America. Figure 2 demonstrates that most breast cancers, breast lumps, around 80% of those biopsied are benign, non-cancers, and around 20% are breast cancer. Although there is a significant percentage, up to 37% of missed early-stage breast cancers are impalpable neoplasms of the breast, and the majority, 76% of the impalpable neoplasms, are benign diseases of the breast. Identification of impalpable neoplasms of the mammary glands can be quite difficult. Well, first of all, they do not cause any complaints, and a woman may not be aware of the presence of a tumor node for a long time. Secondly, due to their small size, especially with a high density of breast tissue, impalpable neoplasms can be easily missed during examination. The slide presents a comparison of the breast cancer screening programs with the cancer detection by presence in developing countries. The difference in breast cancer distribution by stages proves that the use of the organized breast cancer screening mammography allows detecting mainly early-stage breast cancers, whereas cancer detection by presentation, which is opportunistic screening, primarily identifies invasive forms of cancer. Breast population-based screening, where persons are individually identified and personally invited to attend screening, is intended to detect breast cancer at the early stage to reduce mortality rates. Such screening programs based on mammography have already confirmed the efficacy in the reduction of the mortality rate and increased rate of early-stage cancer detection. As you can see from the map, global distribution of the quality of care index on breast cancer is very uneven. At present, only a few countries with a high development index have implemented population-oriented breast cancer screening programs. The global estimated age standardized quality of care index for breast cancer in female in 2020 was 78.7. The estimated quality of care index was highest in high development index regions. The top countries with the highest calculation quality of care index were Iceland, 100, Japan, 99.8, and Finland, 98.8. At the bottom countries were Mozambique, Somalia, 8.2, and Central African Republic, 5.3. Current methods of breast cancer Diagnostics are presented on this slide. Digital mammography has a high sensitivity from 63 to 98% with a specificity of 60 to 90%. Contrast-enhanced mammography has a number of advantages over traditional X-ray mammography and MRI, which is currently considered the most sensitive method of breast diagnosis. It allows to evaluate the structure of the breast, the presence of the asymmetries, accumulations of microcalcinates, and to evaluate the presence of blood flow in suspicious areas. Breast MRI is widely used worldwide as a supplemental instrumental method to the identification, diagnosis, and staging of breast cancer. The MRI is a global survey tool for the identification of breast cancer. It has very high sensitivity, 93 to 100%, at 73, 80% specificity. The use of contrast medications allows estimation of tumor angiogenesis, increased vascularization, which may be a sign of malignant processes in the breast, regardless of the breast density. The use of ultrasound elastography increases the specificity of ultrasound research, enabling a more precise estimation of the size of the tumor nodules. Mammography currently is the gold standard of breast cancer research and the only screening method that has proven efficacy in reducing mortality rates from breast cancer in blinded multi-center clinical trials. In addition to the obvious advantages of screening mammography, which allows the detection of malignant neoplasms less than 5 mm in diameter, detection of 50 per 100,000, that's 0.5% of invasive breast cancers, it has a number of significant drawbacks. In particular, screening mammography produces up to 3/4 of suspicious mammograms. And it also has it misses up to 30% of early stage breast cancers. Therefore, the need for considerable improvement both in sensitivity and specificity of breast cancer screening is the reason for new diagnostic tests that are being developed worldwide. Despite the obvious advantages of the organized population-based screening programs in comparison with breast cancer detection by presentation, or as it's called, opportunistic screening, are available only in a number of countries with high index of development. The main barriers to the organized screening worldwide are the very high cost of governmental screening programs, which have heavy financial burden on the state and limited instrumental capacity. Major disadvantages of existing breast cancer screening mammography consist in a significant amount of false negative and false positive results. The scheme demonstrates that from 1,000 women who underwent screening mammography, 89 women, which is 9%, will have false positive results, 9 will have breast cancer cases, and 1 false negative test, which is missed breast cancer. The probability of having a breast cancer positive mammography is only 9%. To reduce false positive results, additional methods of analysis are required to precisely differentiate between malignant and benign breast tumors. The possible solution is to use a clear CA-62 immunoassay for suspicious mammograms for women above 40 years old. This approach can be effectively used in countries with low, medium, and high index of development. For women with BIRAS 2 and 3, it is recommended to perform a combined breast ultrasound and clear CA-62 immunoassays. Additional methods of diagnostic, including fine needle biopsy, along with the breast ultrasound and the clear CA-62, are recommended for patients with BIRAS 4 and 5 with moderate to high suspicion of breast cancer. The clear CA-62 immunoassay is a one-stage comparative chemiluminescent analysis between the luminescent CA-62 conjugate and the native antigen CA-62 in the serum of patients for binding to monoclonal antibodies immobilized in the microplate wells. Test results are read on a chemiluminescent reader, which is available in clinical diagnostic labs. This slide presents the description of a double-blind clinical study using 488 verified clinical samples, which were carried out together with Sechenov Medical University in Moscow, in which no clinical research group, neither the experts at the Sechenov University, knew the identification of the samples until the analysis of the data obtained. The double-blind clinical study of 488 samples were presented here. The results of the conducted clinical study confirmed very high diagnostic significance of the test of 93.7% for the detection of the early stages of breast cancer, 1A, 1B, and 2A stage, including non-invasive ductal carcinoma in situ. This slide shows the results for all 300 breast cancer patients against all 141 healthy controls and 47 patients with benign breast diseases. The use of the clear CA-62 assay for breast cancer showed maximum sensitivity for 0 stage and stage 1 of cancer, which is 97% sensitivity, which decreased with more invasive stages of cancer. At stage 2 was 85%, and stages 3 and 4, 83%. The resulting diagnostic test accuracy indicators are very likely to detect the earliest stages of cancer, which can be used effectively as an additional method of in vitro diagnostics to complement visual methods such as mammography, ultrasound, and MRI. Detectable levels of various cancer markers at different stages of cancer growth can be significantly separated in time, up to 10 to 15 years. Membrane-like proteins such as CA-62 appear in large quantities from the onset of cancerogenesis on the membrane of various epithelial cells during the epithelial-mesenchymal transition, allowing the marker to enter the bloodstream and being detected at the very early stages of cancer. The maximum expression of ENG-like protein CA-62 is observed at stage 0 and stage 1 of cancer, and it's remained high with the tumor growth. Unlike CA-62, the marker CA-15.3, for example, belongs to the mucin family with high molecular weight, which enters the bloodstream only when cancer cells are destroyed. Therefore, the level of such markers is directly proportional to the tumor growth. This slide shows a very interesting pattern that we noticed when analyzed the obtained results of the two markers measurements. We constructed a regression analysis curve for the results for all patients, and we noticed that all breast cancer patients were crowded into one square located above 10 units per milliliter of the mucin and above 6,300 of the glycoprotein CA-62. This allowed us to consider a combination of two markers to increase the sensitivity of the CA-15.3 marker and increase the specificity of the marker combination to 100%. We have determined the most optimal conditions for the two markers, which are presented on the next slide. For the combined use of CA-15.3 and CA-62 cancer markers, it was necessary to introduce the following mathematical transformations of two independent parameters into one arbitrary value in accordance with the basic principles of the testing process. Samples with CA-15.3 between 10 and 30 have been assigned values for the most sensitive marker, CA-62. Samples with CA-15.3 above 30 units per milliliter have been assigned equation 2 values, and specimens with CA-15.3 less than 10 units per milliliter were assigned values of equation 3. Thus, specimens were considered positive if CA-15.3 was above 30 units per milliliter in a range between 10 and 30, and CA-62 was above 6,300 units, and negative in all other cases. This approach allows consideration of only one parameter for the sample. The slide presents the results of the combination of two biomarkers, which increased the sensitivity of early stages detection to 75% and reached 100% specificity. The use of this combination approach in breast cancer screening for women over 35 years of age will make it possible to identify the very early stages of breast cancer that are well treatable and provide high survival rate. Our latest results presented in Slovenia demonstrated that using the following parameters such as age, CA-62, and CA-15.3 level, the logistic regression analysis allows accessing the likelihood of cancer. For example, for a woman of 54 years old with CA-62 level of 5,800 units per milliliter and CA-15.3 equal to 10.8 units per milliliter, the probability of having cancer is equal to 74%. Binary logistic regression automatically weights any correlation between variables and provides the corresponding statistical significance. Finally, logistic regression assesses the probability of having cancer. rather than just a value in units per milliliter. A possible screening algorithm presented on the slide: patients whose CA-62 level in the transition zone should be observed and blood should be taken for the marker CA-62 for 3 consecutive months during the year for monitoring of the dynamics of this marker to detect the cancer disease or confirm its absence. Patients with CA-62 biomarker level above 6,300 units per milliliter should be referred to instrumental examination for cancer detection. Thus, the clear CA-62 assay reduces the total cost of screening. The slide presents the screening algorithm for women with high breast density. A new optimized algorithm for breast cancer detection will allow to reduce the cost of the health system by eliminating false positive results related to benign breast diseases. Based on the assumption that more than 3 out of 5 positive results of the checkups are false positives, the cost of additional medical examination will be cut by 64%. High risk of breast cancer, according to CA-62 serum level, consists in steadily increased CA-62 level above 5,500 units per milliliter for a long time in the presence or absence of symptoms, significantly increased CA-62 level above 7,000 units at checkup examination, increased level of CA-62 marker above 5,500 during monitoring and remission, and hereditary predisposition. On this slide, I demonstrated the screenshot of the paper that we have on cancer biomarkers just a month ago. Main conclusions of the conducted blind clinical study. The evidence from this study presents remarkable results in regard to the biomarkers combination for stage 0 and stage 1 breast cancer detection. Sensitivity 75% at 100% specificity, which is suitable for the screening for early stages of breast cancer. The diagnostic efficacy of CA-15.3 cancer marker alone can be significantly increased from 20 to 79% by applying a second very sensitive biomarker CA-62 to a reduced to 10 units per milliliter CA-15.3 value in the detection of ductal carcinoma and sinus stage 1 and stage 2 breast cancer at 95 to 100% specificity. 100% specificity in the studied breast cancer cohort samples was obtained at the following combination of the antigens, CA-62 and CA-15.3 antigens, at CA-15.3 between 10 and 46 units per milliliter and CA-62 above 6,300 units, which allows eliminating false positive results. The use of this combination approach in breast cancer screening for women over 35 years of age makes it possible to identify the very early stages of cancer that are well treatable and give patients high survival rate. An assessment of the results obtained using a combination of biomarkers allows that such approach to breast cancer detection has a potential in the nearest future to be integrated into screening programs, current screening programs. And the final, the logistic regression using age, CA-62 and CA-15.3 value assess a probability of having cancer. Thank you very much for your attention and to Scientex conference for the opportunity to present our research achievements.